hopefully you got the newsletter, but I just wanted to reiterate again that we are thrilled, as is the state council, that Laguna River folks, the town of Camden, has received a grant from National Fish and Wildlife Foundation for over $5 million to take out the Montgomery Dam and to work on fish passage there and start the whole process of hopefully restoring fish to the Laguna watershed.
I didn't know that.
Enormous fish.
When did that happen?
Last week, they just announced it.
Yeah, last Friday.
Yeah. Let me see here. They have a coastal resiliency fund, and a lot of people ask, well, you know, I thought the government shut everything down. Well, unfortunately, the government does not control these funds. I forget the source of National Fish and Wildlife, but it's not dependent on an annual budget. So the grant was 5.9 million, and there's another 400,000 in it. So the total project right now is $6.3 million. And they have been awarded the grant. It's been publicly announced, etc. So it's really exciting. The town voted overwhelmingly in favor of that. We were active in that. I got a really nice email to the chapter from one of the town selectmen that helped lead this effort, Allison, and thanked us from being on board from the beginning of trying to make this happen. So assuming this happens, which we think it will, then the next steps will be that the town has already been working with dam owners further upstream to try and figure out what to do there. But none of that would have happened, would not be possible if we hadn't already started with the Montgomery Dam. So really big news. On the other hand, Tim and some folks put together a grant for them for some of our work on here in the Lincolnville area. We did not get funded for that, but that was for how much, Tim?
About $100,000. Right. It was the exact same program that the Camden Dam went in, and I think we just got overshadowed because they're not going to put. As Roy mentioned, these funds were the only funds available. The competition was very fierce. We made the cut right to the end, but I just think that we're not going to put all their eggs into the Mid-Coast Maine, the nationwide program, really. So,
can we reapply next year?
I'm going to talk about that.
Okay, all right.
Yeah.
I don't want to steal your thunder there.
Yeah.
So anyway, that's a big win. As you know, TU National is big on taking dams out all over the country. And as you also know, there's a plan now that's been approved for the National Conservancy, I think, to buy the 4 dams on the lower Kennebec. And assuming that goes through, they're going to take those dams out. So we're making really good progress here in Maine. Steve, do you want to update folks on the Grand Lake Stream Trip?
Yes, briefly, we've gone from 10 guests. Jeff McAvoy at Weatherby's has accommodated us up to 18. Wow. At this point, the 18 spaces are occupied, and we have a wait list of 3. And I'll be speaking with him again in the next month or so. Obviously, he wants to book his cabins, you know, for the folks that are paying $300 a night instead of us paying $79 a night. And but I'll see if there is a chance to get some more, and if so, I make an announcement on that. There'll be details to follow. We'll have some maps for those that haven't fished at GLS, and I'll put together a list of recommended flies. Hopefully, it'll be the Hendrickson Hatch, 16th and 17th of May.
Yeah, thank you very much again for organizing all that. Should be a great trip. So if we get 18 people, or even get 12. That'd be fabulous. It's a big outing. We could put 18 people on that stream. We're gonna. It might be a little cool.
Catch a lot of fish if they're in there.
We'll get up early. Good. Thank you. Did you ever come in?
No.
Almost done here. Trout camp. Again, we have one young man who might be interested here, Rafe. We're looking for candidates. We have to have those candidates' names in to trout camp, folks. By April 1st or so. But if you know anybody who might be interested, please encourage them. They can go online and get an application. There's applications here on the table. I have extras. We want to make sure that we get at least 2 applicants, if not more. And again, that's June 14th through the 18th up in Solon. Any questions on anything? All right, I'm going to turn this over to Tim. Tim is the chair of our conservation committee. He's been the project leader on our main conservation project, which is the Ducktrap River. It's a fabulous river. It may be small, but it's mighty. It has wild native Atlantic salmon and wild native brook trout, and that's a rare thing, very rare. And it's in our turf, and Tim's been leading that effort, so he's going to tell you all about it. So thank you, Tim. Appreciate it. Good to go.
All right. Well, this is going to be a show and tell. We're going to go through some stuff and some slides, but also we have stuff to demonstrate and pass out so people can see what's going on. It's going to be sort of a combination of our conservation efforts and some fishing tips. So we're going to sprinkle it all in here. And I have 10 tips. And I'm sure people in the audience here have other tips. And if they want to speak up, they can. But if they want to keep it all to themselves, that's not really fair. But we won't call them out because they're better than I am anyway on this stuff. All right, we'll get started. All right, let's go. I don't know how to work this. So point this way.
That's forward.
Forward. Okay, here we go.
And then the red dot in the middle is the laser.
Gotcha. All right. You see what we got here? This is a Ducktrap native. What's different between this and a fish you would find on most of the fish you've got on the St. George?
It's wild.
It's wild. It's right. They're not shaped like this to the St. George, being that Purina chow, and they're not the same fish. They're not. They don't, but also they don't have the same genetics as these fish. They probably could not survive in the duck trap because their body shape is not attuned to the duck trap, which over 10,000 years of rising and falling as a spate river, it's shaped the fish. They've genetically adapted to that environment. And you're going to see some pictures in here. So if you have any questions, jump in because you don't want to hear me droning here. So people understand what I'm saying about that? Is that fish, if they spawn in a certain area, they have a small gene pool and variations get passed down to generations very quickly. So favorable variations that help them survive get passed on to their offspring very quickly. That's for salmon and these brook trout in the stream. Okay. All right. What are we protecting when we talk about the duck trap? But one, it's an intact ecosystem of land and water with native flora and fauna. And it's a map I put out here. You can see from Coastal Mountains Land Trust. 85% of it's already protected. It started with the Duck Trap Coalition, which our chapter was a founding member of that. It was called the Duck Trap chapter of Trout Unlimited when that happened. And now it's evolved to Coastal Mountains Land Trust, which has taken over from them. We have a sea-run fishery there. There's eels, alewives, brook trout, Atlantic salmon, sea lampreys, and striped bass. And I'm saying maybe sturgeon because I've seen them at Howe Point, but I'm not sure exactly if they do come up above and into the basin. But I know they're right there at Howe Point looking out. You can see them. But it's not fish though, it's animals. We got bear, moose, deer, coyote, bobcat, porcupine, beaver, raccoon, and muskrat. I've seen them all. And you can too. It is wild, but it's not wilderness. This land is actively reclaimed from a lot of human efforts over the years. And the reason we've got this jewel is because of the conservation. Okay, so again, a long history of human interaction. You know, from the indigenous people. Do y'all know about the Vikings? How many people know that there's a good evidence that the Vikings were at the duck trap? Where does it come from? The Icelandic sagas. Because the Vikings, when they came to America, didn't have a written language. It was all oral. And it's been written down and been studied up. And there's an exact description of Howe Point looking up at Ducktrap Mountain in the Viking sagas. And I think it's some truth to it. At one point, there was actually some structures that were man-made, primitive structures on Ducktrap Mountain. When NBA came, those were destroyed, taken down. They could have been Viking structures. Don't know. But anyway, this is a serious scientists have actually looked into this. And anyway, okay. And I would say besides the Vikings, we have colonists, there's farmers, industrialists. You know, there's evidence of quite a few mills, lumber mills on the duck trap. If you really go around and know where to look, you can see where there was lumber mills at one point. There's also industrialists. This was a gravel pit for many years, a gravel mining operation. And that's one of the key that made the duck trap is so successful is because of the deep gravel bottom, which we're going to get into of the drainage. And it's got nature enthusiasts and conservationists. That's us. We're now here trying to do it. So when you think of the duck trap, what I want you to think of is a maternity ward and a nursery. That's where the fish come to have their offspring, and that's where the juvenile fish have to have a place to grow. That's really the major function of the duck trap reef. Okay. Any questions on that so far? Okay. So what are we protecting? Okay, this is just an example of some native plants you're going to see around the efforts there. And then you've got these little things called brook trout that you're going to see. This is, I would say, an average little brook trout, probably 5 to 6 inches long, born in the river, stays in the river, dies in the river.
Where did you get the bamboo rod?
Henry Marchetti.
No.
Right. As I mentioned, a nursery is not just a nursery of fish. This is a picture I took 2 years ago. Just look it down. In that one picture, what do you see?
Eggs.
You see eggs. They're amphibians. There's actually 3 different species of amphibians in those eggs. If you look really closely, you're going to see stoneflies all throughout there. Right? That shows you how fertile the place is. Okay? It is an unbelievable fertile place for life. And that's just one little shot. Okay, and it's got other things too. The same gravel that makes it good for spawning for salmon and brook trout makes it a great place for snapping turtles because they have to come out of the bank and get in that marly material to lay their eggs. So if you see something that's look like a funny looking rock in the spring, don't reach your hand down and touch that. I guarantee you. That's the head of a snapping turtle. And it's not going to like it if you do that. There's no question about that. Okay. Well, here's another one, a brook trout you can see. And again, the shape of the body adapted to the hydraulics of the duck trap over the years. That's you won't find a fish shaped like that coming out of the hatchery. You won't find it in the St. George. Okay, here. I mentioned how fertile it is. This is we're taking one of our sampling trips this year. This basically was a leftover red from brook trout. You look in here, there's still material. The fish have just come out. You've got 3 or 4 different species of fish right here. You've got suckers, horsehead suckers. You've got dace, and it's hard to see, but there's one little down here that's got the markings on it, over there. That's I can't tell whether it's a brook trout or a salmon fry in there. So it's got a tremendous number of bait fish, especially in the spring, all sorts of food. And that's why we're coming up to fishing tip number 1. Use a muddler minnow. 80% of the fishing pictures you're going to see tonight, fish were caught on this block. 10 or 12, size 10 or 12, muddler minnow. You don't have to get too fancy. And you can take it to the bank. I can tell you, you can use other stuff you want, and your mileage may vary, but that will do the trick. These are not genius fish, they don't have PhDs. So that'll work. Now we're going to go in a few times where you want to do something other than the butter, but 80% of the time you're good to go. Okay.
They're all smart as I am.
Pardon me? Yeah.
They're all smart as I am.
Oh yeah. Yes, sir.
If it's not too much to ask, could you comment on a fishing mat in a small stream?
Okay.
Like
Yes.
the duck trap, because I could see swinging that in a big river, but
Okay. Well, here's the deal. The duck trap in the spring, well, we're going to talk about what times to fish, whatnot, usually has a lot of water. It has more lookable habitat than actually fish could support because the habitat isn't there all year long. So the fish move around and whatnot. And the key is you want deep enough water where you can swing it. That is, you want to fish. I fish mine quarter or actually sometimes almost straight downstream. The key is cast it out, pause, strip, pause, strip, pause, strip paw, boom, they hit on the paws every time. If people don't have other opinions, throw up. You know, this isn't me. It's strip paws, strip paws, strip paw. Boom, and there's another one. It's real different.
All right, that sounds.
Yeah.
Out.
You could strip out and do the same thing. It's on the retrieve. It's on the paws when they hit. And you don't have to be, I usually use 3X tippet. You're not. Any questions on that? There. Okay. And we're going to talk a little bit where to fish for. Okay. All right. Again, we got all sorts of these. These are like creeping dogwood. It's in bloom in the spring. You can see there. Alewives, tons of alewives. They come in normal water levers. They go to all 3 feeder ponds: Coleman, Tilden, and Pitcher. And they have to come up the main duck trap for that. This is they come in. This is in the May, and it's another thing great to go there in the spring, because all of nature knows this is a bounty. I've counted 7 eagles at one time, molting in the lower duck trap, jumping down to pick those fish up, jumping back in the tree because they can't fly, and just eating away. So they know it, and that they're there to eat those elements too. And it's very common to look on a rock and you see scales, and then a little downy feather. It's the eagle has had its lunch. Okay. And another one of the pesky brook trout.
Recording in progress.
You got in there. You got all plants, as Eric pointed out. That was a Jack in the Pulpit. Yeah. Plant. There's all sorts of different plants you can see here. Again, another brook trout you can catch out of the duck trap.
Is that your micro net?
Pardon? Yeah. Well, some of them are pretty good. I mean, let's talk about size. First of all, I'm going to say we've had a lot of programs on fishing the last couple of years on where to go. There's great fishing in New Zealand, Montana, Alaska, other places. You can catch that. It's an 8-minute drive from here to where to catch that fish. You don't have to get a plane ticket. You can do it. 8 minutes right from where you're sitting, boom, to catch that fish.
How big is the gap of your net?
Okay, let's talk about the size. The average size is, I would say the average size of the duck trap fisher is 6 to 9 inches. There is a tremendous number of fish up to 12 inches. There is a lesser amount, but catchable, between 12 and 15 inches. If you caught a 16 to 20 incher, which is possible, then I'll buy you a drink. Because I want to know where you went. So, but I have talked to people, and I believe that, but it's hard to get over that 15-inch mark. The way it goes. Okay. Anybody want to disagree? Yeah.
I mean, I'll interject that most of the time when I go and fish the duck trap, I'll catch at least 1 or 2 that are between 12 and 14.
Yeah.
I've actually taken a tape to one that I thought was 16, but it was 15 and a half.
Yeah.
I haven't broken the 16 mark. I think I've lost the 16 mark.
Yeah. Oh yeah.
But there are some
Yes.
good sized fish if you know where to look for them.
Yeah. So does that answer your questions? I mean, we're being honest with you here. I mean, it's not like a place where you're going to rack up the huge, you're not going to be sitting here like this 2-handed hero shot or something like that. That's not going to happen. Yes.
Tim, how many of these fish do you keep?
Well, I keep 0, but you can keep 2 fish. The state regulations, you see in the brochure, it's fly fishing only, and you can keep 2. So, and some people do, but I'm not going to say bad things about them.
But every one of the fish that you're showing has been released.
Yes. Oh, yes. Oh, yeah. Oh, yeah. Yes. Everyone figured it. Yes. Okay. Okay. More alewives, as you can see, coming to thing. We're going to move a little faster here because I got a bunch of stuff here. Again, there's a real small one here. You can see the par marks. Again, this was probably just like 5 inches. That fish there, but still, it's a beautiful fish. It's a wild fish. Nature put it there, and through our conservation efforts, they're still there. Yeah, there are a few snakes, just a few there, not too many, and they're harmless. More of those brook trout, and there's bobcats. There's a print there. It's hard to see, but it's on the side. That's a print of a bobcat. More alewives that come through the thing there. This one is one of my favorite pictures. This was a really nice fish here. But again, the shape, you can see the shape. I can tell right away that's duck trap fish based on the shape. All right, this is something a little different. You don't see me showing you any salmon, adult salmon. I'll be honest with you, I've only seen 2 adult salmon in the duck trap in all the hours I've spent there. And both of them were in exactly the same place 3 years apart. And I'm talking the exact same spot. So, but what you do see is their evidence, besides their progeny, the par and the smolts, the reds. This is a salmon red. It's hard to see maybe on the trip. This is all cleared out. You can see where the silt is still sitting here. It's been cleared out, and there's a little pile of rocks at the end. That's an active. That was an active salmon red, and that was from last year. So, yeah,
Is that the same one in the same spot?
it was the same spot, but the next year, it was the exact same spot. But Lauren and I went the next year, and it was it. Yeah.
Have any others been observed in other locations in the last couple of years?
No, the water was so low this year, I don't think they did a survey. So, yeah. We're gonna talk about that. Again, some other fish you could get that trap. Again, salmon to give you an idea. So, that's like a 12-inch fish there, but pretty thick. Yeah. There's also turkeys you're going to see. You can see more different. Each one's a little different. It's like a snowflake, a little different coloring, different setup. How they do it. Beavers, you got plenty of beavers there. I always complained about them, but I was wrong. They're part of nature. The fish have evolved with them, and that red you saw would not be there without a beaver dam backing the water up over it last year. So there is advantages to having beavers in the waters. Okay. Again, the typical little brook trout. Okay. There's plenty of deer. Now, what our presentation last week, or what two meetings ago, was on the salters. There's a clear example of one. No question, was a salter. See how it's all washed out, the colors. It's not that the photo is in bad shape. Is that fish just came in from the ocean probably less than 36 hours ago. It's coming to fresh water, and he's just starting to get his colors. You understand what I'm saying?
Effective fly for salters, Tim.
That mother minnow. You can also use others. Harris Special. You could use a Mickey Finn. Mickey Fins I use sometimes, but you don't really have to. You don't have to carry 1,000 patterns to do this. You just got to be able to learn where to go in the right spots. No else. Okay. And also we're going to talk about that. Okay. And we're going to talk about, there's an example of coyotes. I just took my boot print down so you get an idea on what's on the bank. And then another one there. Before I stop, I'm going to give you fishing hit number 2. Mutter minnows number 1. Number 2, this is your most important piece of fishing equipment. It's a thermometer. Why? A lot of the fish, the adult fish, they're salters. They go out into the Penobscot Bay. The Penobscot Bay usually gets temperature in the winter of around 43 degrees. They don't come back into the duck trap until the temperature gets above 43 degrees. Okay? They don't get really active till the temperature gets around 46 degrees. Once the temperature hits 64 degrees, their peak, once it gets above 65, 66, it starts tailing off. Once it hits 68, forget it. This is your important piece of equipment right here. You got to take the water temperature, because if they're not going to be there, if it does have the right temperature. Anybody?
And that's all in the spring.
Well, we're going to talk about that. When should you go? From mid-April through May in most years. Some years all the way through the first week of June. And in some years, but not this year, the last week of September. The water's dropped. If we have normal water levels, and if we get a low pressure system, remnants of a hurricane to come up that last week of September, because the season closes September 30th, I would play hooky from school, or I would call in sick at work, and I would go to the duck trap. Because if you got 2 or 3 inches of water on drop at that time of year, those fish are going to pull in out of Penobscot Bay, and they're going to be right there in the river. And they're going to be prime spawners. They're going to be your bigger fish in peak color. Everybody understand that? I'm giving you all the secrets now. This is like, okay, this is what you want to do. So you want to watch that weather, and I'll explain how you check the water level and all that. But believe me, if it happens the last week of September, go. Okay. All right, good to go there. Okay, let's go there. All right, where to go? We're going to talk about here. Here's the map that you better hear. There's 3 roads that cross the duck trap. There is the mouth at Howe Point. That's Route 1. That's Route 52. There's actually another road not on the map that's up here, right here. I think it's Dickey Ridge or
Dickey Mill.
Dickey Mill, right here. Those are the only 3 roads that cross the whole river. Okay, where most people fish, I don't know, but I'll tell you where I fish. I go into the Camp Tanglewood area, which I think is right here, and fish up and downstream. And then I go Route 50. You can park here. You can fish downstream. You can fish upstream. You can also fish Kendall Brook and Black Brook in normal years. Those are all good fishing locations. Okay. I know some people that I've heard that will tell us about fishing even the lower river down here. But I'm to an old man, I can't get down there, so I don't do that. Okay.
Pretty river down there though.
I'm sure it is, and one day someone will take me down there and show me the hot spots, but that's okay.
All ripples and runs.
Oh yeah, oh yeah. Okay. When I say water level, there is a gauge by the USGS right here at Route 52. It's updated every day on the internet. You can get it. You can get it. Put it on your phone. I got it on my phone. And just hit one button, and it gives you the water gauge. What is the right flow? Well, I'd say below 35 and above 5, maybe 4 if I have to. That's a good flow. If it's above 35, I think it's tough because there's a lot of water running through there. If it's below, then you can only certain spots you can fish. Okay, and that's right there. You can check it every day, an hour. You get an idea whether the river's going up, going down, whatnot. Okay, all right. That was tip number 3, by the way, if you're keeping score. Okay. Okay. Well, this is what it looks like in the winter. We think about it, but this is very important. This is from Route 52. All that water gets frozen and in normal winter, and this is what's going away. When they say global warming or climate change, this is what happens. It's not that it's getting hotter in the summer, it's getting warmer in the winter. That means less snowfall, more rain, more runoff, less ice pack, less ice pack to slowly melt in the spring. It has detrimental effects. We're going to talk a little more about that. Okay. But this is a normal spring flow down at your Turner Falls. It's a beautiful spot. If you've ever been, I don't know if anybody's ever been there. It's really nice. It's really where historically the big salmon pool was, right there below Turner Falls when they were salmon fishing. It's just a very scenic spot. I mean, to be honest with you, the duck trap is so magical that if you went there today and you came back and said, Tim, I saw Bigfoot ride a unicorn. I believe it. It's that magical, the place. It's unbelievable. And it's right in our own backyard. Okay, this is a spring runoff. This is closer to where Tucker Brook comes in. You have to hike in here to get to this spot. Beautiful spot. In the right water conditions, that thing's full of trout. This is again below Kendall Brook, the Kendall Brook pool, which is down there. It's one of the deeper spots in the water. There's always usually fish in there, and there can be some real big ones in there. And that's all we'll say about that. And then there is a wooden bridge which crosses it. This is in the Tanglewood camp. I don't know if anybody knows this. There's a wooden bridge there that goes up to Pitcher Pond. You can see that. Again, it's 3-season situation. It runs clear even in the snow. In the fall, we get really low water. And unfortunately, we got super low water this year. See that? Okay. In the winter, this is right, again, it can freeze up a lot, but it always runs. It never freezes completely solid that I've seen. And this is from Howe Point looking back up the duck trap. And you can see that's a duck trap river, that white stuff on top of the salt water as it's going out. And there's a duck trap river up on the bank as it's frozen going out. So anyway, you get it on here. Okay. This is 2 or 3 years ago at Route 52. in the fall, nice water level. That's this year. We have an unmitigated disaster on our hands. Is that this is one of the lowest I've seen. So it's definitely going to affect the fish population. It's going to knock it back some. We had I can't remember the exact years, but we did have another drought several years ago. In my records, it knocked the fishing back for a year. By 2 years, it all recovered. So I feel confident that can happen. The salmon, they'll knock them back. But remember, they go out to the ocean for 3, 4, or 5 years before they come back to spawn. That's the way nature has programmed them to do that. So one bad year doesn't doom them. Brook trout, they're so prolific. So enough of them will be out in the Penobscot Bay. They'll come back as soon as the conditions are, and they'll repopulate the place. Just takes about 2 years. Good. Again, spring. All right. Here we go. Well, there's actually a few bugs in the duck trap. There wasn't just all minnows. There are some times when you can fish dry flies, and this is one of them. Let me explain this. There's plenty of caddis and mayflies. They're not every section of the river. They're mostly in the rockier sections of the river. And the key is that sometimes you have to fish in the morning, sometimes you have to fish in the afternoon. Why is that? Well, in the spring, the warmer times are going to be in the afternoon. And that's when the peak activity of insects are going to be. That's when these guys hatch, and that's when the fish are active. And later in the year, you want the cooler times, so you fish in the mornings. So you have to. That's what you have to plan into your thing once the thing happens. So that's actually tip number 4 there. Sometimes you fish in the mornings, sometimes you fish in the afternoon. Okay. All right. This is a sample of the little bridge here. And you say, well, what is that? Nothing to that. This hides a very important piece. Almost every one of these little bridges things, these are springs, freshnets. They're all cold water seeps, which go in the duck trap. And there's a way you can tell the cold water. You can put your thermometer there and take the temperature, or you guys are a smart crowd. You probably know more than I do. You study your aquatic plants, you'll know when it's cold water. Do you know what watercress looks like? You want to look for it, because if you see it sitting around in the spring coming out in front, that's cold water. And it helps to catch up on that, so you know where the cold water seeps are, especially late by the time you're into June. Okay. All right, let's talk about conservation. Okay, ongoing conservation. The Coastal Mountains Land Trust has had a multi-year program on eradication of Japanese knotweed. I don't know if y'all know about that invasive plant. It's been going on for quite a while. It's pretty successful. They've knocked it back quite a bit. Fly fishing only designation, catch and release of brook trout is not really true. We try to foster that, but you can keep 2. Keep 2 by the state. No fishing for Atlantic salmon, and public education is what we've been trying to do. Okay. All right. Mention a warming planet. I'm not going to read all this through you, but basically, as I mentioned before, stream flow is going to be affected because we're going to have less snow in the winter, earlier runoff, rivers going to dry out quicker. This is a textbook example of what's happened. We have a drought. You can have drought type conditions and such. It's also going to affect the forest compositions. Basically, the hemlock trees, which is a lot of hemlock trees in the duck trap drainage, and beech trees, they're going to be under stress. And climate models show they're going to be replaced over the years by more hardwoods, which is going to change the whole forest canopy and the amount of water vapor that's lost in the air and all the type of stuff. So that's what we're up against. Okay. So what have we done? What is our club? I'm talking our club here, done to do this. One, we've got the Ducktrap is qualified as a TU National Priority Water Program. What does that mean? It's one of the 200 places that Charter Limited is willing to put money in to sort of save, because it's worth saving. And this is an example of Charter Limited National buying this pump here, which we're going to show you a little how it works, for $2,200, and we get to use it for free. So they bought that just for us, and the other chapters too. But we did a summer water temperature study in 2023, and what we used. You can pass these around. A couple of these people could see what these are called hobos. I don't know if you've ever seen these things. the little, basically a coin battery goes in here. You program them with a computer to take the water temperature. And we programmed ours to take the temperature every hour on the hour. We put them in the stream as we went along. And we left them in there from June through August, then recorded the results to determine the cold water sources and whatnot. And it's old technology. These were used on the original rock ramp project on the Georges River, and still works. And if we have another project want to come up with, we still have them that we can use, and it works fine. Okay, we're going to go into that. We did an invasive species study, the eDNA, in 2023 before we had the pump, where we went out and sampled at 14 locations to figure out the eDNA of what invasive species are there. We put an automated angler survey in, which is ongoing, which I'm going to ask you, if everybody's got a cell phone, when I get to that slide, I got a QR code. I want you to take a picture and scan it, because if you go to the Ducktrap, I want you to fill out one of those forms. That's very important. Okay. We supported the Coleman Pond outlet remediation in 2017, which fixed the outlet at Coleman Pond going into the Ducktrap. We participated in the DMR red counts in 2023. We've supported the Penobscot Nation's grant request for doing work on the Ducktrap, predominantly the Pitcher Pond out, which they're trying to get money for to fix that. It's a terrible fish play. We put a grant application in for Black Brook under the National Fish and Wildlife Foundation, which we didn't get. The Montgomery Dam did get it in our area. So that doesn't mean our grant was well received. It was, and it doesn't mean that our priorities doesn't need to happen. It's just we didn't get the money. We'll see. And we did an eDNA monitoring program in 2025, which you're going to see the results for, which we use this. And we got a grant from TU of Braces Stream for additional 2 years of eDNA study. And I've got the sign up list here for anybody who's interested to help on that project as we get along. I'll go into that. How big was that? $4,000. $4,000. Yeah. And you may see some people here. They're in the audience here today. So, okay. What is priority water? Basically, it means that TU National is stepping up. I mean, we wouldn't have gotten the pump if it wasn't priority water. It's clear as a day. And we have resources helping us, and Lauren helping us on projects. She helped put together. She did the whole grant application for the Black Brook remediation. So we're getting support for our dollars going to the club. Okay.
To show the significance of the priority water designation, how many other streams or rivers in Maine have that?
Let's see. There's 1, 2, 3, 4, 5, 6. I think there's 8 in Maine. We're one of the 8 in Maine. One of the 8 in Maine. One of 200 nationwide, 8 in Maine. Yeah. Okay. Now, why the Ducktrap? What's important? Again, it's one of the only 8 rivers in America supporting wild Atlantic salmon. They have the salmon in the Ducktrap have a unique genetic profile. There's different. They've done studies on it. There's the Sheepscot had salmon. The Penobscot had salmon. The Kennebec had salmon. And the Narragansett and Denny's rivers had salmon. They're all unique to those specific locations. The fish, yes.
And isn't the Ducktrap the only one of the 8 that was never stocked? So
Correct.
That's the original DNA.
The original DNA. They've never been stocked. And when the salmon were put on the endangered species list, the National Fish Hatchery at Craigbrook was established to be a salmon breeding thing. There was no Atlantic salmon from the Ducktrap put in that program. The Ducktrap was left out, and so was Cold Brook near below Bangor. The ones in Cold Brook are now believed to be extirpated. We're the only ones left standing. So there's no plan B for our Ducktrap salmon. If the conservation goes away, they're going to die, fail out, and that's going to be it. There's no way about it. We are the last line of defense for the salmon in the Ducktrap river. Okay. Let's see. You can read the thing, but basically, this is a great opportunity where we can actually bring young people in and help and let them learn about the conservation, because it's right in their own backyard, and it's a unique thing that can't be recreated. Okay. All right. I mentioned the thermal study. This is one we did in 2023. you can see, we did, I think, 1, 2, 3, 4, 5, 6, 7 different areas. Basically, it comes out that this ponds, Pitcher, Coleman, and Tyndall are, they're heat sinks. They capture heat in the summer, and the water comes over. There's no bottom release dam or anything. It just comes out of the top into the river. So that's hot water which comes to the duck trap. The duck trap cold water fish to survive, they have to either go to the ocean or find thermal refuge within the river system. And where's the thermal river in the river system? These little tributaries: Tucker Brook, tributary north of Route 52, and the Black Brook on Slab City Road. That's the major 3 cold water resources that we've got in the river. Am I clear on that, or that basically those fish, they're dependent upon those sources of cold water? Yes.
So those obviously don't come from the ponds, those are coming, that's water coming from somewhere.
Correct. Yeah, Black Brook comes from off of the backside of Furlock Mountain. Tucker Brook comes from off of Greenacre Road, groundwater seepage, which comes in there. And this little thing north of Route 52, it comes out of the ground. I think this is a spring. It's unnamed tributary there. It's a spring that comes in. There are other small seepages and springs in the lower river. Don't get me wrong, but they're not major. I mean, you'll find them if you look around in summer. You'll see them. Go ahead.
I'd interject that there are some places on the duck trap as well that there are these really deep troughs that are carved out of clay, and those correspond to where you'll see springs in the forest beside. So there are some deep channels that are 5 or 6 feet deep that in the summer the fish, I think, just lay on the bottom of those like slow troughs, because there are places where these channels get really deep that I have to imagine the temperature variation in it is significant.
I agree. And there's some of these places, they don't warm up. I mean, there are a few places that you'll be walking along even in June, and there'll be ice, little ice on the side there where it's so cold in the back. Oh yeah, they're dependent on those things.
Yeah.
Yes.
What's the deal with Kendall Brook? It's warm now.
Yes.
Isn't it? How did they ever have a fish hatchery on Kendall Brook?
I don't know. That's a good question. Well, I believe they pump water out of it. Well, I do. I believe it has a deep aquifer around there, and I believe they pump water out of it. It's under the gravel. I believe they pump water out. That's my understanding. I forgot about into it. Then it would come down as semi-cold water the rest of the stream. When Ducktrap fishery originally had a fish hatchery there. Yes. Yeah. Yeah. But I believe they pump water out of a pump. I mean, there was an artesian well on the Tanglewood property where they pump water out many years ago too, when the Civilian Conservation Corps was there. And I believe that still has some cold water, because it looks like, I look at the vegetation around there, and it looks like cold water vegetation to me. So, yeah. But again, these are minor. These are the major spots. Okay. Okay. All right. We did an invasive species study like in 2023 using eDNA, where Dan and I basically hiked into 14 locations and we survived. Did it, carrying water bottles in and out. And this is one of the results. Very, very important, is that these are we tested for 4 invasive species: largemouth bass, smallmouth bass, black crappie, and northern pike. We did not find northern pike. We did find these other species, and you can see where they're coming from. Largemouth bass and smallmouth bass are coming from Tilden Pond, Coleman Pond. Largemouth and smallmouth bass are in the area. From down here, from Pitcher Pond, largemouth bass and black crappie coming in. You can actually easily figure out where the fish are coming from by the eDNA study. So what we did after that is we put on a catch and kill campaign for the bass, and don't believe me, we didn't get everything, I'm sure of that, but it did knock the population back, because I'll go into that in a minute. Okay. Okay. We do a duck trap angler survey, and we've done this now for 5 years. I just didn't update the thing, finish it this year. And what have we learned? Okay, Water thermometer is very important, which I talked to you about for success. You need to get the water to the right temperatures. And as you can see, once that water temperature gets in the 50s, success shoots way up, 50s and 60s. My God. I mean, you should be very confident about catching a brook trout once water gets in that range. If it's 70, forget it. You're not, it's gone. And if it's too cold, lower, it's just, you're not going to have as good a fish. So that's why this is important. Everybody put this as a stocking stuffer. It's not expensive. You can get one. There you go. Okay. All right. And this, if this is the QR code, scan it with your phone. It'll put you and save it. And it's an automatic Google survey form. And every time you go fishing that direct trap, it's answers 5 or 6 simple questions. And it really makes a difference because this data has correlated unbelievably well with our eDNA study. So, yeah. All right. All right, Coleman Pond remediation. There's some people's here. You may recognize the pictures. Some really ugly guys are in here. They're not here to defend themselves, but obviously Dan Daly and Terry, they were their major head person, and they were. That's basically putting in a culvert on Slab City where Coleman Pond comes into the Ducktrap River, into Black Brook. That was done in 2017. This is what we want to do to Black Brook, the next tributary. We did salmon red cow in 2023. There's the biologist from the state looking in there, and there we are cogitating what's going on. How's it going? All right, this is little information I left in here on the National Coastal Resilience Fund. What we applied for, same thing as the town of Camden. And this is what the past things they've done in Maine. And this was actually the preliminary study, which they did at Camden years ago, that's what we were applying for this year, to get a preliminary study done. We put together this. This is, get an idea, this is the drainage of what we're talking about. This is Slab City Road right here. That's Coleman Brook. This is the thing we fixed in 2017. This is what we want to fix next. It controls all this cold water coming off Rock Mountain back in here. Okay. So that's what we want to fix. You'll see a picture. I'll show you what's wrong with that. And actually, we do this investigation. These two things here are actually colonial remnants of colonial mills that were put on the Black Brook back, in the 1800s, I guess. But they've been breached, and the fish can get through there. Okay, this is Black Brook right down Slab City Road in flood conditions. This is why it needed to be fixed. This is the state's solution. These 3 huge pipes here flowing water. Unfortunately, right now there's now a 5-foot drop from those pipes to the water in the sun and the regular rime. So no fish can go up that. Nothing can come up. Nothing. They're hung. They can't get up that. It's called a hung culvert. That's what we want to replace with an at-grade crossing so the fish can go up in and out. That's our goal. We didn't get the funding for this. The need hasn't gone away. We'll be looking at other things with the current funding situation up in the air. If it was 2 or 3 years ago, I'd say we were slam dunk, but I don't know. We'll have to see. I'm not a politician. Okay. All right. This is what the star of the show. Whoops, I'm running a little late here. Get going here. This is our eDNA project we did in 2025. And this is what we use. This is what we've got money to continue. I'll pass this around. This is the whole process. This is the pump. We have to carry this in, and Eric's got a pack basket, which just fits in exactly right. So we carry this in. Someone puts on rubber gloves, takes this filter, which is about 25 or 30 bucks, attaches it to the end of the hose here. Well, you can demonstrate it because you did it. Come on up here and demonstrate it.
I don't remember.
Anyway, basically, it just goes in and it goes in the water and it pumps till we get this is full. Then we have to take it out of the water, reverse it, blow the air out, put it in the bag, seal it. It's good for 6 months. We can keep it on the shelf for 6 months before we send it to a lab to get analyzed, gets marked. It's super simple. It takes the way we did it last year is that we sampled 4 weeks out of 5, 6 locations each time, so it was 24 samples, and it took about 3 hours to do the sampling each time. So it was 4 times we had to do 3 hours of sample, and we took volunteers. You could do it once or more, that would be fine. I got a sign-up sheet here if you want to get interested in. I'll be sometime this winter. I'll be putting an email list out once we start looking at what the schedule is and whatnot, and it would contact people, see if they're interested. That's all you got to do. You don't, no commitment tonight. All you do is you get on the email list, and I'll send you an email. But it would probably be, go ahead, sir.
What does our chapter cost for each of those filters that we buy?
A case of 25 was roughly $500. So yeah, that's what it costs. So we have enough funds to basically, it's going to cost us almost $6,000 to do the study for the next 2 years. We got 4,000 already. We're still looking for another 2,000 over the next 2 years. Then it'll be totally paid. So still looking for that. Yeah. Yeah.
Want to mention
Yeah.
that I volunteered with him all 4 of those sessions. And, I think it's good to be aware that in the Tanglewood section, there's a lot of long hiking in your ways and whatever. There's lots of routes. I mean, it can be tiring. So if you can't walk a couple miles in your ways, then maybe it's good.
Yeah.
But it's beautiful. Oh, it's beautiful. Get even more. You got to.
That's right. In fact, that brings you to tip number 7. Volunteer for a conservation project. You're going to learn a lot more about the fishing as we're walking down the river. You'll get a lot more tips if you do that. All right. And you'll learn more about the river. It's good to learn. Volunteer for a conservation project. Okay. All right. Well, that's what we've got. Let's see here. Let's see. Well, what do we find out in our study? And why are we doing this? Okay. Well, we did 6 locations. You can see 2, 1 above Route 52, 1 right below Route 52, 1 around Black Brook Confluence, 2 around Cuttlefoot Confluence, and 1 at the near Turner Falls. And basically, because of the high water we had early in spring, we missed a week. We had to skip a week. So by the time we did the last testing, the water has started to warm up. And you can see we had 100% early in the year. And then lower in the river, as we tailed off a little bit on detecting the brook trout. About 75% of the time, 100% of the time, we got it over here. So early, basically, there's a good healthy population of brook trout. Okay. Salmon, different story, but this was amazing here. At 50% of our test, we found brook trout here. I mean, Atlantic salmon. This is right below Route 52. And then none above. So what does that tell you? They're right. This is where this is where they are. The eDNA did not drift down from above because we didn't detect anything up there. The river flows this direction. So this is where the fish were. They weren't detected down here. So this is a highly critical area we've identified for the Atlantic salmon. And why that's important, I'm going to show you a little bit later. Okay, and then we did find them down here where Kendall Brook comes in prime areas too. So this was all good news. That was good news because you could talk to some people and they said there's no salmon left in the Ducktrap. Well, that's not true. You know, we have indefutable evidence that there is. And besides catching them ourselves and releasing them, we have scientific evidence. Okay. Okay. What is this? What happens when you take these samples? We're in the river taking the samples. And I know this is the high science crowd, maybe not here. Basically, the University of Maine takes these samples, takes the information out, runs them through a special machine, has a comparison DNA of the species you want to test, and they put this enzyme on it, on our sample, and it multiplies it exponentially. So how many times it has to multiply before it gets a detectable level gives you the strength of the sample. So in essence, like golf, you want a low score. So normally you have to repeat the process from 20 to 40 times before you get a detectable level. Most of ours came in 20 to 38 times. They were very high. It was very undisputed that we're not false positives. They were getting the right results. Okay. again, this is a scientific study. I'm not doing a good job on this. I know. But I mentioned the thing has to be replicated. So if you get it 20, it shows that's it's a higher concentration of DNA. If you go in at 30 times, it's a little less. If you have no eDNA in the sample, you don't get any amplification. There's nothing to amplify. So, okay. Okay. So what are we going to do now in the future? One, we're going to continuing our eDNA monitoring that we're doing now. We may change the locations. I'd like to have a Zoom call this meeting for anybody that's interested, so we can talk about the sampling strategy. Should we have sampled the exact same locations? Should we switch it up? What some thoughts are on that? We got to do that. Another thing we need to do is we've identified at least one area in the watershed that the salmon are using right now. And we know that area needs to be reforested because it's lost its forest canopy, mostly from beavers, natural aging of trees, and stress on the beech trees. We need to contact and come up with the state forester, the coastal, and this happens to be in Coastal Mountain Land Trust property, so there's no problem getting their permission to restore it and do a tree planting effort to get that doing. But that's something that I know nothing about. We'll have to go to TU National, get more information, and do an overall plan. Not this year, maybe next year, after 2 years of study, we'll be able to do that. Okay. Thermal imaging drone to identify more cold water refugia. The main TU has a thermal imaging drone and a very expensive software where we can fly it over the Ducktrap watershed, potentially identify cold water sources. Problem is, you have to have line of sight, and really it only works after the leaves fall off the trees. So this would be something we'd have to do fairly early in November up here. Maybe next year we would plan for that. Okay. Drone use for identification of Atlantic salmon and brook trout reds. This is something that's actually used in Montana, and we potentially could use it here for in the duck trap. I'll show you something here in a minute about that. And then last but not least, this came out. I don't know if you saw this in the New York Times just 2 weeks ago. They now can track individual monarch butterflies by cell phone technology, putting a tag on them. When I saw that, I immediately wrote down the information and checked them out, and they're waterproof. So we potentially could do a project on that. We can't tag the salmon because we don't have a license, but we could tag the brook trout. And if there was enough cell towers in the coverage drainage, we'd be able to track the brook trout. So that's something for the future because we have poor cell coverage right now in the duck trout. So, but that's we're always thinking here that George River trout are living.
I think it's starlings.
Well, that's true. We could check on Star. We're always thinking. Okay. And that's about it. Could you close up? Just hit the end slide. So I hit one more thing. Well, no, go right there. Hold it right there. Go. If you could put your cursor on a little thing and hit that. Oh, hit that dot. It's the open link, yeah. And then make that big, yeah. This is a video put together by David. David and I went out earlier this year and we looked at using drones on the Ducktrap. And this is the exact area where we had the high concentration of Atlantic salmon and our eDNA study. As you can see, this is the river right on the Ducktrap. Beautiful, beautiful river, beautiful section. It's prime habitat.
What month was that?
Pardon me?
What month?
This had to have been in April. I think we were in April. We did this. Yeah. It was just, I mean, it's a full stream. And this is exactly where, we did our sampling in May, but we took the video before that. Yeah. And you get an idea. That water's clear enough that if they were, potentially we could do a red count by looking for the bright spots in the gravel. Something to think about. That's what they do in other places. So not expensive. Just got to do a little thought to it. Put together. And that gravel is the key, buddy. That's spawning gravel. Okay. Okay. So I'm going to give you I'm low on my tips here. This is tip number 8 on your fishing. If you catch more than one salmon par on a location, I ask you, move on to a different beat. Because the salmon par grouped together in school, they're stupid. You catch one, you go back and another, you catch another one. No, you don't need to do that. Just move on to a different beat. You won't, the brook trout won't be there. There'll be salmon par. So just give the salmon par a break. Okay.
Tim, how to differentiate between salmon, par, and brook trout?
As I go back, when it's finished, we'll go back to slide number 2. Yeah, go back to the original slideshow. Yeah, go to slide number 2.
Is it made into fork tail?
No, it's the coloration.
Give me a second.
Yeah, go back to our original presentation. Go back. Go to slide 2.
Strong.
Yeah.
R marking on the
Yeah.
Atlantic salmon.
Yeah, so. Yeah, well,
Small salmon look very similar to a small brown trout.
back to
Only
Yeah.
with forked tail.
slide number 2, way up at the top. Yeah. Slide 2.
Yeah.
That's courtesy of Patrick, that picture, the background there, that is, that's a salmon par. Right there. And you see, it doesn't look like anything like the brook trout you've seen before. The coloration looks like a brown trout. Okay.
White background, black spots.
Yeah,
Yeah.
Bring it to your hand immediately and release.
at least that's all you gotta do, yeah. So,
They'll generally have little red dots
yeah,
above the lateral line.
Yeah.
There's usually a yellow tint to
Yeah.
they look a lot like a small juvenile brown trout, only if you look at the jaws, their jaws don't extend past the eyes.
correct.
So like a brown trout's jaws would
Yeah.
extend past their eyes. Also, just anecdotally, most of the places that I catch the salmon par are on dry flies in ripple sections. So if you're using Tim's Mother Minnow or my white
Yeah.
streamer, you're probably not gonna find too many salmon
Yeah.
par, but if you're using a dry
Yeah.
fly at the head of pools, right where the ripples come in, that's where the
Yeah.
par are.
Yeah. Atlantic salmon like the more oxygenated water, no question about that. So, okay. All right, last tip I have is have fun and file a report. Because those reports that's gonna help us, because if the no salmon are detected for 12 years, they're deemed to be extirpated. And we know that's not the case. Okay, so it really helps if we get the data now. So I've droned on quite a bit. I hope that it's not too boring. And I hope you got an idea of what we're doing here and what we're looking for. And I appreciate it. Even if you have any interest, put your name and email down here, and I will I'll put you on a list, and you can decide later if you want to help. That's it. Yeah.
Questions for Tim?
Yeah.
This is our big project, multi-year.
Did I pass it?
Sorry, I'm sorry.
Oh, yeah, got it.
One little announcement on the Ducktrap project.
Sure.
So Tim put together the thing that was, in one of the emails, a whole presentation. I submitted that to the, Maine Natural History Observatory. They put out 4 times a year, they put out a digital magazine. It's a small organization, but they want to publish that, so that will be in the next issue. So we're gonna get some advertising for the Ducktrap Unlimited and for our shipping.
What's their website?
What's the name of the organization?
The organization is the Maine Natural History Observatory. And MaineNaturalHistory.org is the website. And I've contributed to them for a while, but they do some really nice field guides. They're working on a field guide for 2 weeks now, but they realize that they're in trouble because there's way more species of seaweed that look exactly alike than
Other questions or comments for Tim?
You should feel if you need help fishing the duck trap, just give me an email.
Yes, you took me, and I can't fix it. Let's do the raffle then. All right. Dan, you wanna, how about ask Rafe if he pulled the ticket? You willing to do that, Rafe? Maybe pull the tickets there out of the hat? Yeah. All right.
Anybody else have
And
any?
what we do is, as you remember, we pull a ticket and you get about 5 seconds to get up there and take your choice. Then we call another one.
That's
So
right.
it goes pretty quickly.
Yeah.
And you could,
Yeah. All right, last 8 numbers are 890. 890.
What I do, but take it. Yes, sir, right here. Make sure I can read your name and email. There you go. Yeah, it'll be sometime
889.
February, I'll get it.
Steve, all right.
Yeah.
Sort of where one.
It's 880.
880.
No.
880.
Correct.
Okay. Good job.
Pennsylvania.
Yeah.
Great job.
Put out 8 players.
Yeah.
Out eggs.
Eight, nine,